Welcome, everyone, and thank you for virtually joining us for the latest version of the Variant Effects seminar series. My name is Diego, and I'm joined by Mireia Seuma, who is the other member of the VESS organizing committee here today. These seminars are made possible with support and a lot of help from Lara Muffley and Alex Hopkins, among several others, and our sponsor, the Atlas of Variant Effects Alliance. So a big thank you to them. Before jumping in, I have a few housekeeping items to mention. Each talk is about 20 or 25 minutes, so there should be some time for a few questions at the end. Please post these questions at any time to the Zoom Q&A or chat, and we'll read them out to the speakers at the end. If you have additional questions after the end of the seminar and you're an AVE member, feel free to visit our seminar series Slack channel for more discussion. You can catch the seminar today and previous presentations that have been recorded on our CMAP YouTube channel. And finally, we're posting updates on our Twitter account, so be sure to follow us there at Variant Effects. This has never been an issue, but as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. All the details of this seminar series can be found on our website, including a regularly updated schedule of upcoming speakers. I believe the next VESS is May 3rd in the 9 to 10 a.m. slot. If you've enjoyed these seminars, then be sure to register for the Mutational Scanning Symposium, which will be held online and in person in Toronto on June 13th and 14th. The website listed here has all the details. Just keep in mind that abstracts are due the 9th and the early bird registration deadline is the 15th, both of this month. Okay, so without further ado, we have an excellent lineup for today's presentations. Our first speaker, Shawn Fayer, is a genetic counselor and PhD candidate at the University of Washington, co-advised by Doug Fowler and Leah Storita. His work focuses on the clinical integration of multiplexed functional assay data and generation of multiplexable assays in pluripotent stem cells. The title of his talk is Closing the Gap: Systematic Integration of Multiplex Functional Data Resolves Variants of Uncertain Significance in BRCA1, TP53, and P10. Shawn, feel free to start sharing your screen and go ahead and start whenever you're ready.
Right. Thank you, Diego, for that introduction. And big thank you to all the organizers for the invitation to give this talk today. This talk is really an example of how advancements in technology, genomics technology development can drastically improve clinical genetics. And to give an example of what I mean by that, I'm just going to take us quickly back in time to about 2014, when I was doing my genetic counseling training in cancer genetics. And at that time, when we saw patients who had personal and family history of breast cancer, what we would do is offer sequencing for 2 genes, BRCA1 and 2. And when we sequence these genes, we would find that about 4% of patients, represented here in red, would have a positive result, meaning we found a pathogenic variant that explains their personal and family history of cancer. An additional 4% of patients would have variants of uncertain significance when we sequence these 2 genes. So these are represented now in gray. And this means that we found a variant in either BRCA1 and 2, but for these patients, we don't know if that variant is a pathogenic disease-causing variant or just benign population variation. And the majority of patients in this cohort would actually get negative results back, meaning we sequence both genes and we don't find any variants in the genes. So clearly we have an enriched population, but we're not finding very many patients that have pathogenic variants. As our sequencing technology improved and we identified more genes associated with hereditary cancer syndromes, we added more genes to panels and improved the diagnostic yield of these panels. Now today, with some of our modern panels where we sequence between 40 and 80 genes associated with hereditary cancer predisposition, we find that about 10% of patients in this hereditary breast cancer cohort will have a pathogenic result or a positive test on that sequencing panel indicating that they have a hereditary cancer predisposition. So this is great. We're now identifying more patients that have pathogenic variants, and we can now, counsel these patients on what they can do with this information in terms of following up with advanced screening and surveillance, as well as preventative options. What also occurs when we test for more genes is we find many more variants of uncertain significance. So we went from about 4% VUS rate to now 40% with these expanded panels. And this is actually problematic because now the majority of patients where we're returning a variant to, we're telling them we don't know what this variant means. Furthermore, when we return a variant of uncertain significance, we tell these patients, this is information that is not going to change your medical management in any way. And we don't recommend, any of your family members be tested for this variant. And the best we can tell them is, please check back with us in a year or 2 years, and we'll see if there has been an update to your clinical variant interpretation. And when I say variant interpretation, what I'm referring to is a specific set of guidelines set out by the American College of Medical Genetics for trying to bin sequence variants on clinical genetic testing into one of 5 bins: benign, likely benign, uncertain significance, likely pathogenic, or pathogenic. In order to bin a variant in any one of these bins, we use multiple levels of evidence from the patient's phenotype to segregation information. So do variants in the gene segregate with disease and families, population incidences of your variant, and then probably the 2 pieces of evidence that we're most familiar with on this call, the computational predictive models and functional assays, where we actually measure the impact of variants. And for each of these pieces of evidence, we assign a weight or a strength of evidence for either a pathogenic effect or a benign effect. And the strengths go from strong down to moderate and down to supporting. And to give you an idea of sort of the difference between these levels, you could have 5 or more pieces of supporting evidence for a pathogenic impact of a variant. But in the absence of any moderate or strong evidence, your variant is still going to be called a variant of uncertain significance. Whereas if you had 2 pieces of strong evidence for pathogenic effect, then you'll interpret that variant as pathogenic. When we actually make these interpretations, what we're saying about a variant is its expected chance of being disease causing. So when we call a variant benign, what we're saying is this variant has a less than 5% chance that it is disease causing. If we move up to likely benign, we're saying there's a between 5 and 10% chance a variant is disease causing. And clinically, we sort of use these interchangeably. Anything 10% or below, we're saying this is benign, this is not disease causing. Conversely, if we're calling something likely pathogenic, we're saying there's a 90 to 95% chance that that variant is disease causing, and pathogenic is greater than 95% chance. What you may have noticed, though, is that the greatest chunk is this middle class, uncertain significance, where any variant that is between 10 and 90% chance of being disease causing is called a variant of uncertain significance. And for this reason, most of the variants that are interpreted clinically fall into this VUS category. If we zoom out and take a look at all of the variants across disease genes that have been uploaded to the ClinVar database from clinical genetic testing, what we see is that nearly 75% of all variants are variants of uncertain significance, represented in gray. Now, this is a problem that's actually been growing over time. So doing the same ClinVar data poll from 2015 annually, we can plot the variant classifications and see that the definitive classes of benign and pathogenic only modestly increase over time, whereas the VUS nearly exponentially increase over time, represented by this gray line. Now, the reason for this is actually quite simple. We're just sequencing more genes. As we sequence more genes, we find more rare variants, and rare variants are more likely to be variants that have limited pieces of evidence that we can put towards that variant interpretation and have a higher chance of being VUS. Now, while we can't do very much about the rarity of these variants, what we can do is generate functional data for variants at scale. And the way that we do that is through the generation of multiplex functional assays. I know we're quite familiar with these assays on this call, so I'll just touch on this very briefly. In a multiplex functional assay, what we do is we take a cell line of interest, and then we have a gene of interest that we want to functionally annotate. So in that gene, we introduce variants such that each cell has only a single copy of that of a variant in that gene of interest. And then we put these cells in pooled through a functional selection, and then sequence variants at the end of this functional selection and score them based on the relative fitness of variants. And our hope is that we can now use these sequence function maps to help inform clinical variant interpretation. When I joined the Fayer and Streeter labs, there actually hadn't been a study looking at the actual clinical impact of multiplex functional data. So we decided to ask the very simple question, what is the clinical utility of multiplex functional data in reclassifying variants of uncertain significance? To answer this question, we took previously published multiplex functional data for the tumor suppressor genes BRCA1, TP53, and PTEN. And I'll touch on these assays individually, starting with BRCA1. We used the data from the saturation genome editing assay that was led by Greg Finley in the Shindri lab here at the University of Washington. And what I'm showing you here are variants of known clinical impact that were measured in the assay, plotted by their function score along the x-axis. Variants that are represented in red and orange are pathogenic and likely pathogenic variants that were measured, and variants represented in blue and light blue are the benign variants that were measured in the assay. And as you can see through this distribution, we have a really nice separation between these two classes of variants. And because of this, integrating the BRCA1 function scores is actually fairly straightforward. Things get a bit more complicated when we look at TP53, where we were able to pull data from 4 different functional assays. 2 of these assays looked at any loss of function variant in TP53. And then the other 2 assays specifically looked for dominant negative variants in TP53. And then finally, we used 2 different multiplex assays for PTEN, one that looked at variant abundance in a mammalian cell model, and one that looked at PTEN phosphatase activity in a humanized yeast assay. The clinical data that we used in this analysis came from Ambry Genetics, who shared their variant interpretations for variants in BRCA1, TP53, and PTEN that overlapped with the functional data that we had available. And as you can see from this clinical data, the majority class of variants on an individual gene level are also variants of uncertain significance. And so we applied the multiplex functional data to try to reduce the amount of VUS in each one of these genes. And the way that we did this was through a 3-step process. The first is to simply collect and then combine the multiplex functional data for clinical integration. The second step is to calculate that strength of evidence for each assay. And so again, the strength of evidence is just a measure of how predictive a functional assay is of the pathogenicity of a variant. And then finally, in step 3, we take those evidence strength codes and apply them to the clinical variant interpretation with the evidence that Ambry had already used in their variant interpretations. So I'll walk through this step by step, starting with BRCA1, since it's sort of the most simple case. So after collecting the data from the BRCA1 assay, it's always a good idea to do a sanity check. You know, is this assay measuring what you expect it to measure? And so a way to do that for BRCA1 is to plot the nonsense and the synonymous variants in the assay. So we know that loss of function is a pathogenic mechanism for BRCA1 variants. So if this assay is accurately measuring loss of function of BRCA1, the nonsense variants plotted against the synonymous variants should give us a nice separation. And so that's what we indeed see here. So I'm plotting the nonsense variants in this purple-pink color and synonymous variants in green. And the vertical lines represent 95% cutoffs for each of the distributions. So now anything in the pink shaded area to the left of the nonsense distribution are going to be called functionally abnormal variants because they behave like the nonsense variants. And anything to the right side in the green shaded area will be called functionally normal because they behave like the synonymous variants. And so the next step is to then actually calculate the strength of evidence we can apply to variants that fall into the functionally abnormal and functionally normal classes. And the way that we do that is by, once again, plotting function scores along the x-axis, but now for variants of known clinical effect. So these are, again, the pathogenic and benign variants from the ClinVar database. And the vertical lines now represent the cutoffs for both the functionally abnormal and functionally normal classes of variant. We then calculate a statistic called the odds path, or odds of pathogenicity, for each functional class based on how well the assay actually distinguishes between known pathogenic and known benign variants. So for any variant now that falls to the left side of the distribution based on this validation and is called functionally abnormal, we calculate the odds path, which comes out to 52.4, which corresponds with strong evidence for pathogenic effect. And on the opposite side, for any variant that's functionally normal in the assay, we calculate an odds path of 0.02, which corresponds with strong evidence for benign impact. We then take these evidence codes and now move on to the actual variant interpretation with these evidence codes. And for BRCA1, we were able to reclassify 49% of all the US and Ambry's data set to either likely benign or likely pathogenic. So 49 variants of uncertain significance were more definitively interpreted as likely benign, represented by the shaded or the striped blue and gray portion of the plot. And 5 variants moved from variant of uncertain significance to likely pathogenic. I just want to take a quick pause here to really drive home the point that each one of these reinterpreted variants represented a patient or a group of patients that had a VUS through their genetic testing through Ambry, who then had a updated sequencing report generated and sent to their ordering provider to return to them now an updated variant interpretation. So 49 of which were told your VUS is now considered likely benign and not thought to be causative of a familial cancer syndrome. And 5 individuals or families were found to have now a likely pathogenic variant and now have access to, again, advanced screening and surveillance, as well as preventative measures like preventative surgeries to reduce their chance of developing cancer. And now they can also offer testing to potentially at-risk family members who would want to follow up with screening, surveillance, and preventative measures themselves. So that was sort of the simple case for BRCA1. When we move on to TP53, as I mentioned, things got a bit more complicated. Now, instead of one assay, we're trying to evaluate 4 different assays. And if you look at these distributions of the variants of known clinical impact across each of the 4 assays, what you can see is that we don't have as clean a separation between the pathogenic and benign variants as we did for BRCA1. When I calculated the odds path for any one of these assays individually, that odds path calculation was very limited to supporting evidence and thus limiting each of these individual assays in their clinical impact. However, we reasoned that because these 4 assays looked at, were performed in different cell lines, or 2 different cell lines, used different functional selections that looked at different pathogenic mechanisms of TP53 variants, we could combine the scores of all 4 of these assays with a probabilistic classifier to come up with a unifying prediction of TP53 variant functional effect based on the score across each of the assays. So we trained a Naïve Bayes classifier on all of the variants that I'm displaying here on the screen. So all of the ClinVar variants that were pathogenic, likely pathogenic, benign, or likely benign. And then in our cross-validation of this classifier, the classifier predicted 39 variants to be functionally normal and 123 variants to be functionally abnormal. When you overlay the actual interpretations of these variants, all 123 variants predicted to be functionally abnormal were actual pathogenic variants, and 32 of the variants predicted to be functionally normal were benign variants, with 7 misclassified pathogenic variants as functionally normal. So our predictions weren't perfect, but we can still calculate our odds path and our strength of evidence from that odds path. So for any variant predicted to be functionally abnormal by the classifier, we give that strong evidence for pathogenic effect, because we do a really good job of predicting pathogenic variants. And we give moderate evidence for any variant predicted to be functionally normal, because we do slightly worse at predicting benign variants in the functionally normal class. So taking these evidence strength codes forward, we reclassified the VUS in Ambry's TP53 data set, and we're able to move 69% of the VUS to either likely benign or likely pathogenic, which again is a very significant piece of the puzzle for the VUS and TP53. Very excited by how well this combination of multiple assays work for TP53. We're excited to use the same strategy for PTEN, because again, we have multiple functional assays that are measuring different functional impacts of PTEN variants. However, as you can probably see from these two plots, there are only 2 likely benign PTEN variants known, or at least there were only 2 known benign missense variants of PTEN at the time of the paper. And so this one limited the odds path that we can calculate for either of these assays. So we couldn't apply functional evidence based on odds path alone. Additionally, these 2 benign variants limited the ability to train a classifier because we don't have enough variants to train on. So instead of applying the same logic we did for TP53, we reflexed to the ClinGen variant curation expert panel guidelines for PTEN variant interpretation. And we're still able to reclassify 15% of the VUS in Ambry's data set with the multiplex functional data. But again, the data here were limited by the availability of these control benign variants. But now zooming out across all 3 genes, I hope I've demonstrated to you that multiplex functional data does have a pretty substantial clinical impact in resolving variants of uncertain significance in clinical genetic testing, but that utility is variable by gene. And to really drive home where that limitation comes from, it's really in the limitation of control variants to validate our assays. So on the left side, I'm showing you the control variants for BRCA1 and the validation based on those control variants. We have ample pathogenic controls and ample benign controls to validate that assay. And we were able to reclassify a significant chunk of the VUS in Ambry's data set. On the right, we only have 2 benign control variants for PTEN. And so we were limited in how many VUS we could reclassify for this gene. Now moving forward and looking forward, there are more genes that are like PTEN than there are like BRCA1. So more clinically relevant genes that have a limited number of control variants that are at least available in the ClinVar database. So generation of variants of known effect is going to be a major important factor moving forward. And so this will take, in my view, collaboration across the clinical genomics community, from clinical geneticists to testing laboratories and to developers of functional assays, to identify, share, and validate variants of known effect, so we can improve the amount of known benign variants for some of these genes to validate our assays. And then finally, we do also need to consider expanding assays into different phenotypic spaces. So everything that I showed you today was data for tumor suppressor genes measured in predominantly cancer-derived cell lines. So these cell lines are great for functionally annotating tumor suppressor genes. However, they're very limited when you want to look at genes associated with hereditary heart disease, for example. So moving into IPS differentiable models will be very useful in this space moving forward. And with that, I would just like to quickly thank the Fayer and Streeter Labs for their support in this project, specifically Doug and Leah for shared enthusiasm for variant interpretation. A huge thank you to Ambry Genetics for sharing their variant interpretations and helping with finalizing our variant interpretations in the paper. And a thank you to our funding sources. And I would be happy to take any questions.
Great talk, Shawn. Thank you. So questions I think are just starting to trickle in. There's one here in the chat from Daniel Yang, who writes, great talk. So Daniel had a question about the P10 MAVE. Given how limited they were, the number of benign variants, did they study synonymous variants? And if so, did you include that in your odds path analysis?
Yeah, so that's a good question. So both P10 MAVEs did validate their assays with synonymous variants and known pathogenic variants. So while they both measured quite a few synonymous variants, those synonymous variants don't actually go into the odds path calculation. And the reason for that is in the odds path calculation, what you're really measuring is the ability to actually change the amino acid sequence in a protein of interest that is expected to be benign and not change the functional impact, and actually reliably measure that in the assay. With a synonymous variant, you're not expecting there to be any functional impact or any impact in your function score. So you don't actually use that in the odds path calculation, but it was important for both of those assays to actually just validate that they had an assay that was working as they expected it to work, but it didn't go into the odds path calculation.
Great. So we have another question here from Moz Dawood. Says, awesome talk. Can you speak to your confidence in downgrading a variant of uncertain significance to LB? Do you feel negative evidence in one functional assay, or in the case of P53/4 assays, is enough to remove the uncertainty?
Yeah, so I think that's a very important question, and a point that I do want to drive home with this talk, and that I probably didn't speak to in enough detail, is using the ACMG criteria for variant interpretation. We very rarely use only one piece of evidence to make a variant interpretation. So that would be a evidence strength that I didn't even touch on, which would be the very strong class, which is super rare. But in the case that I showed, we rely on multiple pieces of evidence to make a variant interpretation. So if we move a variant to the likely benign category, we're using multiple pieces of evidence back towards that benign assessment. And importantly, we don't have evidence leaning towards pathogenicity in those cases. So I would say I'm actually quite confident in those interpretations because we used, the more holistic use of all of the evidence available to us, and we're not relying solely on the functional data to make that interpretation. The functional data just gave it that extra boost to get over the edge towards that likely benign classification. But that is a good point.
Great. So a couple more questions here. So Fritz Roth says, thanks. For any of these maps, is there a way to identify subsets of functional scores that are more or less reliable? If so, where there is a sufficient set of reference variants, BRCA1, TP53, have you tried calculating odds paths separately for different subsets?
Subsets, so like subsets within a single gene. I think what the question is getting at, and Fritz, please correct me if I'm wrong, is you could have, like, say, different functional domains across a gene, and maybe your assay works really well in one domain and less well in a different domain. And can you zoom into that one functional domain where you have very reliable score prediction? And yeah, subset of variants with more or less reliable functional scores. Yeah, so I think so. And for BRCA1, both functional domains that were measured were actually super reliable. So we didn't really have to do that. But for TP53, it is a bit more interesting because within the DNA binding domain of TP53, all of the assays actually perform quite a bit better. So if you zoom into the DNA binding domain, your odds path actually increases. And it's when you look at sort of the ends of the protein with the oligomerization domain, where things are a little bit less clear, and you have some of those less penetrant but still pathogenic variants. And so that can dilute out the odds path. For the purpose of this paper, we just looked at everything. We didn't make those dissections across the protein. But I don't think there's any reason that you can't do that when you calculate your odds path, especially if your assay is specifically looking for a specific functional impact that would be specific to a certain domain. So I hope that answers your question.
Yeah, thanks again for that awesome talk, Shawn. There's a couple questions in the chat. Maybe you could just respond to those directly, but we should maybe move ahead.